So, looking at the aspects of genetic engineering, right, as I said, right, what we'll be looking at is how, um, well, the general principles. What are the main things that we look at in genetic engineering at this level? What is, what are the processes specifically, right? And generally, right, we're going to be also looking at CRISPR as an example through the syllabus, right? And we're going to be CRISPR in sense of bacterial defense, right? And then we're going to be looking at the implications of genetically modified organisms, right? So let's look at this. So the principles of restriction enzymes now. We know that restriction enzymes now are what we call the molecular scissors, right? They cut DNA. They cut genetic material, right? So what we have here is a recognition sequence, right? Restriction enzymes are proteins that recognize specific sequences in the nucleotides in DNA, right? Which we call the recognition sequence or recognition site, right? So these tend to be palindromic in nature, and this means it's read the same way forward as backwards, right? And here we have E. coli, here we have Bacillus species and a Haemophilus species. And there are different restriction enzymes, right, that we get from those sources. So these bacteria produces these restriction enzymes that cut at these specific sites. All right, that should be lovely, right? No lagging. Hopefully there is no lagging at all. All right, so that's what's happening there. So the cutting specifically now, once a restriction enzyme recognizes its specific recognition sequence, it will bind to the DNA there, and it will either cut in a it either will cut near or within the site itself, right? And it would either cut in what we call like a zigzag pattern, right? Or to create sticky ends, or what we call a blunt end, a straight line to create like a blunt end, right? What's happening here now? We know that sticky ends specifically, right, are what we have when enzymes cut DNA in a staggered manner, that zigzag manner, the staggered manner, right, leaving some overhang parts of one-stranded DNA, right? And the one-stranded DNA can easily anneal or like attach itself to other DNA fragments. That's why I call it sticky. They can easily attach to other DNA fragments. And then we have blunt ends now that are originating from restriction enzymes that cut DNA in just a straight line, right? A straight line. So let's have a look at the restriction enzymes, well, the different ends, right? So here we have BAMH1, right, which will cut in a staggered pattern to produce sticky ends, right? And then here we have K3 or HA3, K3, right, that we have creating a blunt end or blunt ends. All right, everything is fine so far? Lovely. So everything is cool. Because it's gonna get more complex as we go on, right? So applications of restriction enzymes now, we know that we use them in a wide range of microbiological techniques. For example, in restriction fragment length polymorphism, right, RFLP analysis, or what we normally know a little bit more closer to home, DNA cloning, right, and genetic editing, right, gene editing, right. Example with CRISPR Cas9, right? So by cutting DNA at specific sites, researchers can actually manipulate and study DNA sequences, right? Because it's gonna be DNA is a very, very long molecule. So if we can only cut out a piece of DNA and study it, right, that will be very advantageous. Principles of ligase enzymes now. We know that ligase enzymes now will actually act as the molecular glue. So what are we talking about now? So for DNA repair, ligase enzymes, right, are responsible for catalyzing the formation of phosphodiester bonds between adjacent nucleotides, right? So it's responsible for that establishment of the bonds there, right? Joining fragments as well. So in microbiology specifically, right, DNA ligase is used to join or paste DNA fragments together. So this is particularly useful after DNA has been cut by restriction enzymes and the researcher wants to insert DNA fragments into a plasmid or another DNA molecule. So after we cut using restriction, then we paste using ligase enzymes, right? So the process specifically now, the ligase works by catalyzing the formation of the phosphoester bonds between the 3' hydroxyl group of one nucleotide and the 5' phosphate group of another nucleotide, effectively sealing the nick in the DNA backbone. Right, then we should have gone through like the release structures, right, of DNA, etc. And we're gonna have a brief look at it again, right, later down. So applications now, we know that ligase enzymes are essential in molecular cloning techniques such as recombinant DNA technology. And we're going to be looking at a lot of recombinant DNA technology because that's the main thing that we're going to be looking at in this syllabus. All right. So they're also used to repair DNA in living organisms, so you can actually do that. All right. So here now, right, but if we cut something with BAMH1, right, we're just cutting the same thing, right? We basically have a fluorescent label, right? So the gene for fluorescent label, right, with BAM H1 site, right? And what we do is cut it now, and then in order to attach it back now, we can use ligase. All right, so that's really the recombinant gene technology, how it really works. So restrict, then ligase. All right. So recombinant DNA, now we know that recombinant DNA refers to the artificially engineered DNA molecules that are produced, right? So once we cut different parts of DNA and then glue different parts of DNA together, we're going to have what we call our recombinant DNA. And organisms that are derived from derivatives of recombinant DNA, right, primarily within the fields of agriculture, medicine, and biotechnology, we call these things genetically modified organisms, right? So we have that there. So the creation of recombinant DNA typically involves the use of selected specialized enzymes, such as restriction enzymes to cut the DNA at specific sites, then DNA ligase to join the DNA fragments together, and these enzymes enable precise manipulation of DNA sequences, facilitating the construction of complex genetic constructs, right, for a wide range of applications, right? So we can create multiple variants of different recombinant DNA structures, right, that do different stuff. Alright, we're gonna take a quick look at CRISPR, right, specifically. What CRISPR really stands for is the clustered regularly interspaced short palindromic repeats. That is what CRISPR stands for. And it's really a bacterial defense mechanism against invading genetic elements such as viruses and plasmids. Right? Why we really study CRISPR is because if we understand, right, how bacteria really figure out how to defend themselves from viruses, specifically using CRISPR to really break down the genetic material and then store information from the genetic material, we can figure out how to combat viruses. So there are vast medical applications for CRISPR. Right? So generally what we do, right, how CRISPR works, right? We have the acquisition rather of spacer sequences. So when a bacterium is infected with a virus, right, or other foreign genetic material, it incorporates small segments of the invader's bacteria into its own genome, right? So these segments are known as spacer sequences. So what the bacteria does is take in some of the enemy DNA or the invader's DNA, right? The acquired spacer sequences now are integrated into the CRISPR array, which is a region on the bacterial genome, right, on that that bacterial plasmid, right, that consists of repeating DNA sequences interspersed, right, with space sequences, right. So we're gonna have those DNA sequences in the middle of the spacer sequences, right. So we have those repeats are all spaced out, right. That's what it's saying. Each spacer sequence corresponds to a specific viral or foreign DNA sequence, right? And what we have happening now, we're gonna have transcription and processing happening now. So the CRISPR array is transcribed into a long RNA chain called a precursor, right? CRISPR RNA, right? So what we call a p-CRISPR RNA, right? This pre-CRISPR RNA now is then processed into smaller CRISPR RNAs by cellular enzymes, right? So what we're generally looking at is something like that. So we take the bacteria, what it does is it takes in the virus DNA, right? The viral DNA rather. It integrates it into its CRISPR mechanism, and then it starts to produce RNA, right? Reading the DNA and starts to reproduce RNA, right? So the CRISPR RNA now, right? Each of the CRISPR RNA forms a complex with the CRISPR associated proteins, what we call Cas, right? So that's where we get Cas9 from, CRISPR associated proteins, right? Forming the CRISPR Cas complex. So the CRISPR RNA serves as a guide to recognize and bind to complementary sequences in foreign DNA or RNA, right? And what we tend to do now, right, with the Cas complex, right, what it starts to do now is surveys the bacterial cell for foreign genetic material, right? When it encounters a sequence matching the cRNA, it binds to the DNA or RNA first, right? And then the protein then cleaves the DNA or the RNA, destroying it, right? So preventing infection or replication of invading genetic elements. So that makes sense, right? So first, a virus, like a bacteriophage or something like that. We're gonna speak about stuff like that in Unit 2 biology. It attacks the bacteria, and then we know that the virus, what the virus does is input genetic information, right? So it can replicate, right? So what the bacteria does is takes some of that genetic information, right? It reverse transcribes it to create RNA, and we bind that RNA to a CRISPR to a protein complex, right? And what we basically do now, right, the protein complex now will find and read any of that viral information. Because remember, we already took a copy of the virus. So we'll look for any other copy of the virus floating around in the cell, and it will start to break it down. So that's how it really defends against viral infections, right? And if we understand how CRISPR works, we can create different types of medical applications, right? So we can actually fight against viruses more effectively. Because viruses can, so bacteria can actually fight off viruses way better than we can, right? Then we have memory and adaptation now. The bacterial cell retains the memory of past infections by storing spacer sequences corresponding to the invaders in the CRISPR array. This allows for the bacterium to recognize and mount a rapid defense previously encountered, against previously encountered viruses or foreign genetic material, right? So that is what happens here with CRISPR, right? The syllabus under Objective 4.1 asks us to speak about, right, editing genomes with CRISPR. So we can use CRISPR to actually edit genomes, right, and create genetically modified organisms. So that is generally what we're looking at, okay? All right, so that's really CRISPR down. Are there any questions or concerns though, thus far before we move into the rest of the processes that we look at?
Sir, I'm not understanding this. Could you please like simplify it or something?
Okay, so to simplify it really, so what's basically happening here, right? If we look at how CRISPR works, so how the system basically works is that what we're gonna have is some amount of virus invading, right? So some amount of virus is gonna invade, right? The bacteria, and the bacteria is gonna take in the some of the information, right? Some of the genetic information from the virus, right? So what happens now is that once it takes in that information now and has RNA, right? Takes in the RNA from it now, it's going to combine with the Cas9 protein, right? And then the complex now that is formed with the RNA and the protein, right? Now attaches, right, to the genomic DNA sequence, right? So it now actually creates an entire complex per se, right? And now what the protein will do now is that anytime it finds any type of DNA that is similar to the DNA to the RNA that it has, right? If the RNA reads the DNA and it's the same, it's going to break down the DNA. So basically what it's saying is now, let me think of it like this, right? It's like you're told to how would I say this now? So any enemy, right, invades, right, the cell, right? What the cell basically does now is take DNA or take information from that enemy, right? So take the information from the enemy, right? And now what it does is use a protein, right? So what it does now is actually takes the protein and the information, right? And the protein will go around the cell looking for the virus, the enemy, right? So what it will do now is look at the information and sees that if the information it took from the enemy is the same, right, that means that what we're looking at is the enemy. So what it will do now is break down the genetic material, right? So it works similar to something like that, right? So let me see if I could find a diagram of CRISPR that would work lovely for you guys and put it into the PowerPoint. So I think a diagram would be a little bit more advantageous. So let me get a diagram. All right. Are there any questions or concerns while I just go for that diagram so I can show you guys a diagram? Mm, doesn't seem to be any. All right. So let me stop sharing the slide real quick so I can edit it and then show you guys what I'm talking about. How CRISPR really works. Mm. Trying to give really a simplified understanding of how it works. I don't want to go into all the major reactions and stuff like that. So I'm trying to just say, takes in DNA, reads it, wherever it finds something similar to it, it destroys it, right? Something like that. No, that's not what I want. All right. So I'm going to reshare now, and then we'll have a look at the diagram and see how that works. All right. Did I stop sharing? I think I should have. I should have stopped sharing. Okay. Let's go to the thing and see if we can figure it out. Well, we should be able to figure it out now. All right. So everyone can see the slide. Can we see the slide? Okay, Divine can't see it. Interesting. Oh, we can see now. All right. So this is really the guide DNA. So the guide DNA is a part of the guide RNA is a part of the genetic information that we took from the virus, right? Because technically the virus has an RNA. So if a virus attacks the bacteria, it's gonna take a piece of the genetic information, right? And then it's going to actually attach it to its own genome, right? So what we're gonna have now is a Cas9 protein now is attaching it there, so we can see it there now, matching the genome sequence now, right? So what it does now, while it's reading any DNA or anything like that, right? What it will do is disrupt the gene itself, right? And insert a new gene, right? So the gene has a new sequence. So what it will do is break apart the DNA or insert things into DNA to cause frame shift mutations, right? So it basically neutralizes the enemy, if that makes sense. Does that make a little bit, is it more clearer with the diagram? So basically, if you match up with the RNA, because the RNA is taken from the enemy, right, from the virus, right? Therefore, if you match up with the RNA, it basically reads as viral DNA. So it's going to be breaking apart the DNA or inserting a new gene into the DNA in order to neutralize it. Does that make sense? So it searches through the cell, it reads the DNA, and if the RNA now tells us that, oh, this is
Sir, can you
viral
go
DNA,
over it, please?
that is what happens. Go over it. So really, what's happening?
Yes, sir.
Okay. So remember, right? Let me just go over it. Like, let me just simplify it, right? Really, because the syllabus asks us to mention it, right? So what we're saying, virus attacks bacteria. Bacteria takes piece of viral gene, right, and attaches it to its own genome. Full stop. All right. That's generally what we have to know. All right. The CRISPR array now that is within the entire bacteria now, right, is transcribed into a long RNA molecule that is now broken up, right? So those RNA molecules, right, are really reading for viral information. And we just use that there, right? It's a guide, right? And then now that guide RNA attaches to the Cas9 complex, Cas9 protein. So it's now a complex, right? And now, what the complex will do now is go around the cell surveying genetic material. It goes through genetic material now, right? Going through it, reading through the DNA, scanning through it, running through all the DNA, right? All the genetic information, right? And if it senses, right, any amount of enemy RNA or foreign RNA or foreign DNA, right, it will destroy it. That is what's happening. So basically, I took some of your RNA so I can find where um you are, and then when my RNA binds now, that's how I know that this is it. This is foreign information, right? And then I break it, I destroy it, right? And that's how CRISPR works.
So, sir,
Go ahead.
I do unit 2 biology also, and this kind of sound like immunology. Does it relate back to that?
Yes, it is. Right. This is basically what
So,
your blood cells do.
does the CRISPR have antigen? So when I say it took piece of the foreign entity, basically it's like antigen and the whole receptive binding thingy to figure out where, so it can like engulf it and like get rid of it.
Well, not really engulf it, but the antigen technically it is an antigen. Technically, but like it's an it's a viral component. But the thing is, no, it's different from what happens in humans, but it's similar because it is bacterial immune response. That's what it is, right? And we're just looking at genetics to really figure out like what's happening here. All right. So the syllabus just asks us to
Okay.
mention it. Lovely. So if there are any more questions or if there's a possible question, then we'll look at it. But in the interest of time, we're gonna move on. All right. Go ahead, Ivan.
So sir, the long RNA that the thing is transcribed into, that's the guide RNA, right?
Yes.
Okay,
So
sir.
it uses that RNA to find any other foreign piece of information so it can destroy it. All right. So
Can you go back to the diagram, please?
diagram here. There are other diagrams now. So if you Google diagram, you'll figure out that one works better for you. All right. Then you should.
Okay, sir.
All right. So let's look at this bacterium, right? We're now going to be going into really how we create recombinant DNA, right? And it really occurs in these steps here. So the first step, right? We're gonna remove the plasmid from the bacterium itself, right? And then we're going to know react it with the restriction enzymes and ligase, right, and stuff like that. We're gonna now modify the plasmid, right, and then allow it to re-enter. It's reinserted back into the bacterium, and now the bacterium will actually show, right, express the new genetic information that it has, right. But I wanted to get into a little bit more of how it generally works, right. So plasmid extraction. Plasmids are circular DNA molecules, right, found naturally in bacteria and are often used as vectors, carriers, vectors in genetic engineering. The first step is to extract the plasmid of interest from the bacterial cell, and this is typically done using plasmid extraction kits, right, or by employing biochemical techniques to isolate the plasmid DNA from the bacterial cell lysate, right? So this is something that we would have done, well, that I would have done, right? But like, if you're going to go into like first year biochemistry, etc., in your molecular biology labs, which you will do in your first year, you're gonna be extracting a lot of plasmids, right? So here now is the first thing we do. We extract the plasmid. Then we do plasmid editing now. Now we're gonna figure out, what do we want to remove from the plasmid? What do you want to add to the plasmid? How do we do that? Now that we remove the plasmid, what are we going to do to it? We're going to edit it now, right? So once the plasmid is extracted, it can be edited by introducing desired changes or inserting new DNA sequences. This editing, right, can involve various techniques such as restriction enzyme digestion, then ligation, or you could even have site this directed mutagenesis, right? But we're not going to talk about that specifically. It's in the slide if anybody wants to go and read about it. It's really interesting. It's really cool. That's why I included it. It's really cool. But what we're focusing on at Cave Unit 1 is just restriction enzyme digestion to cut it, right? And then ligation. So we use restriction enzyme to cut it, right? Then we insert the DNA that we want to insert into it, and then we glue it back. Basically, it's like a box. We open the plasmid, we put what we want in the plasmid, and then we lock back the plasmid. Alright? That's what's happening there. Now we have verification of editing now. After editing the plasmid, it is essential to verify that the desired changes have been successfully introduced. Right? So it's really a fail-safe measure right here, the fail-safe measure. This can be done using techniques such as DNA sequences, restriction enzyme digestion, or agarose gel electrophoresis to analyze the size and integrity of the edited plasmid, right? So agarose gel electrophoresis, that's a general technique that we tend to use. It's really it's not really an outdated technique, but it's an aged technique, right? It's something I have done before. So if we want to go through how agarose gel electrophoresis is done, right, then probably we can do another go through at some later time, right, within the day or tomorrow, or another day, of how we actually do agarose gel electrophoresis, right? If it's possible, I could find it if I have a biochemistry manual that I still have on me, right, where it actually goes through the procedure. And I actually do have my biochemistry manual on me, so let me look through it. This is chromatography. Where is the agarose gel electrophoresis? Alright, so what I'm going to do in the WhatsApp group for the agarose gel electrophoresis, I'm going to send a picture of the lab write-up and lab procedure for agarose gel electrophoresis. And I'm gonna show you guys what we generally do at UE anyways, and how we use BAM H1 and HIND, right? One, in order to actually cut stuff. How we do genetic modifications and stuff like that. I'm gonna see if I have the information with me, and then I'm going to send it to you guys. Usually done in microbiology though. Right? So that is that. Transformation of bacteria. So what this is basically saying now is that we're going to try to use calcium chloride, right? We're trying to use chemicals now or other methods to now increase the permeability of the cell membrane. There are different methods to do it, right? But one method really uses implementation of calcium chloride in order to make the cell membrane more permeable to its surroundings, right? And then now we have to select the transformed bacteria. So how do we know which bacteria responded to this now, right? What we'll then use now, right, to ensure that only the bacteria containing the edited plasmid is selected, that the transformed bacterial cells are plated, right, onto selected media containing antibiotics and other selective agents. You guys will look at that in higher levels of microbiology, right? And now the plasmid used for editing typically carries a gene for antibiotic resistance. So usually what we're gonna do now, we're gonna make sure that it's resistant. And what we're going to do now, right, when we grow these bacteria on petri dishes, right, the culture, right, we're going to induce or we're gonna introduce antibiotics, right, or other selective agents to the bacterial culture to kill off any bacterial colonies that are not the ones that were edited. That's how we do it to now select our bacteria that we use, right? And then now we now grow it, right? We continue to grow our isolated bacterial colony, right, to express the recombinant DNA, right? So the transformed bacteria containing edited plasmid are cultured under conditions that promote the expression of the recombinant DNA, right? So we usually use bacterial colonies. We use colonies of Bacillus aureus. We use colonies of Escherichia coli, right? We use a lot of different colonies, right? Pseudomonas, right? We grew a lot of different colonies and stuff like that, and we generally know what to do, right? What conditions to put these colonies under, right? In order for us to increase the proliferation of our genetically modified bacteria and now see how the recombinant DNA is expressed, right? That is generally how we do this amount of recombinant DNA creation, right? And this is a diagram really summing it up. So first now we use the promoter, we have the promoter and the target gene there, right? We have the original, we have generally here, right, the antibiotic resistant gene there, right? We have this, right? All we do now is create the recombinant plasmid, right? And we know what that is, right? That is through our restriction, right? Enzyme. So we cut the enzyme, we cut the plasmid, right? So in this case, right, we have an all purple ring with blue here, right? What we do, we cut the plasmid, right? And then we insert the plasmid. So you know that the new gene, the target gene, is now a part of the plasmid. And now we use ligase, right, to ligate it to glue it back. And then we transform now the transformation of the plasmid, right, into the host cell. So we transform the host cell, meaning that we increase the permeability, then we insert the new plasmid, and then what we do now is grow the transformed bacteria, right, on on a selective agar plate. What does it mean by selective, right? It means that it will now, what we'll do is grow it in a media that is suitable for our new GMO, right? But it will kill off everything that is not GMO. Notice that there is some amount of antibiotic resistant gene present, right? And then what we'll do now, only the bacteria, right, with the plasmid that we modified will grow due to the antibacterial or antibiotic rather resistance, right? So that's what's happening there. We have our gene, we open it using restriction enzymes, we add our new gene and we close it using ligase enzymes. Then we transform our cell, right, by adding our plasmid, right? We increase permeability, then add the plasmid. Then we selectively choose to grow the one that is modified, right, due to its antibiotic resistance properties. And that is what we do. The same thing that we use for E. coli and the insulin gene, right? So we attach a gene that creates insulin, then we insulin in the entire process, and then now the bacteria will continue to produce insulin. So it's stuff like that, right? This is generally how the entire process goes. 5 key steps. All right. So that is that. We isolate the plasmid. Remember, we isolate the plasmid, right? So plasmid extraction, right? We isolate it, then we edit it using restriction, then add in what we want to add in, then we ligate it, we close it back, then we make sure that we verify that it's editing. We know no, this is just a verification process, right? To make sure that we now have our new DNA sequence that we want, right, through agarose gel electrophoresis. Then we use transformation bacteria. We increase the permeability of the membrane so we can now insert our new plasmid inside the bacteria, and then we selectively grow our new bacteria, our no recombinant DNA bacteria, right? And then we look for its expression, right? So that's generally what we're doing. So this is a different, but look at E. coli and what we really look at it using for, right? So that's what's happening. So I went through it in depth. That is what's happening here. All right. Any questions or concerns before we move on? Well, let's end this clip here and then we'll move on to gene therapy.